In the last 2 lectures, we studied about T-cell tolerance. Let's now study B-cell tolerance. We know that both origin and maturation of B cells occur in the bone marrow. B cells have their own antigen-specific receptors, which are known as B-cell receptors. Later, B cells migrate to peripheral lymphoid organs where they encounter antigens and get activated. They proliferate and differentiate. The effector B cells are known as plasma cells that secrete antigen-specific antibodies. We have also studied that our immune system generates a population of B cells having a vast repertoire of antigen-specific receptors. During generation of these receptors, some receptors capable of recognizing and binding normal constituents of human body are also generated. We also know that receptors and cells that react against a person's own tissues are known as self-reactive or autoreactive. And the components to which they bind are called self-antigens. If such mature autoreactive B cells get activated, the result will be very damaging to our body. This is because these autoreactive B cells will produce potentially harmful antibodies against own molecules. Such antibodies are known as autoantibodies. This may further disrupt normal functions of the body, damage host tissues and cause autoimmune diseases. So, it becomes crucial for our immune system to prevent maturation of such autoreactive B cells. In this video lecture we will study central B cell tolerance. Let's begin. As we said, the origin and maturation of B cell takes place in the bone marrow. Immature B cells in the bone marrow express B cell receptors on their cell membrane. Our immune system screens each of these immature B cells for autoreactive B cell receptors. The autoreactive B cells are either eliminated or inactivated. Now, there are 2 questions here. First, what is the source of self-antigens in the bone marrow? And second, how autoreactive B cells are distinguished from other B cells. Immature B cells in the bone marrow come in contact with a wide variety of self-molecules or self-antigens. These include the cell surface molecules expressed by stromal cells, hematopoietic cells and molecules circulating in the blood plasma. The recognition of the autoreactive B cells depend on the strength with which B cell receptors of immature B cell bind to the antigens in the bone marrow. Most of these antigens encountered by immature B cells in the bone marrow are multivalent. Recall that multivalent antigens are most effective at cross-linking B cell receptors. These multivalent antigens include glycoproteins, proteoglycans and glycolipids present on the surface of cells in the bone marrow. Most of these self-antigens in the bone marrow are also found on all body cells. If immature B cells do not interact with any of these self-antigens or the interaction is weak, they successfully leave the bone marrow and migrate to the peripheral lymphoid organs. If immature B cells react very strongly with the antigens in the bone marrow, in other words, there is a strong cross-linking of B cell receptors on binding to multivalent self-antigen, their further development is arrested. These cells are the potential autoreactive B cells. They are retained in the bone marrow. Central B cell tolerance uses 3 main mechanisms to induce self-tolerance. These are receptor editing, clonal deletion, and Anergy. Let's understand them further. Receptor editing is the mechanism of establishing self-tolerance in which the antigen receptor of developing autoreactive B cell is modified, It is based on the fact that immature B cells are still capable of rearranging their immunoglobulin genes. This ability of receptor editing is provided by the multiple V and J gene segments in the light chain locus. Suppose, this is an immature B cell, having B cell receptors, and this is a multivalent antigen. Let's say, it is a cell in the bone marrow having identical cell surface molecules. If this immature B cell is self-reactive, or in other words, there is strong cross-linking of B cell receptors of this B cell, an intracellular signal is generated. As a result, this B cell continues rearranging its light chain loci. Self-reactive light chain is deleted. It is replaced with another light chain sequence. If the new B cell receptor generated by this light chain gene rearrangement is non-self-reactive, this B cell continue to develop and become a mature B cell which migrates to peripheral organs. If the new B cell receptor produced is still self-reactive, then further light chain rearrangements are tried by the immune system. During these attempts of receptor editing, some B cells never form a non-self-reactive antigen receptor. Moreover, a time comes when no additional light chain gene segments are available for recombination. Such B cells undergo cell death by apoptosis in the bone marrow. These B cells are then phagocytosed by macrophages. This process, in which developing autoreactive B cells are eliminated by apoptosis, is known as clonal deletion. Now, monovalent antigens are also present in the bone marrow. These are mostly soluble proteins that carry only one copy of an epitope. Self-reactive B cells that bind to monovalent self-antigens are made unresponsive by the central B cell tolerance. Their further development is arrested. This state, in which immature self-reactive B cells become inactivated or unresponsive to their specific antigen, is known as anergy. Anergic cells do not die immediately. Instead, anergic B cells are allowed to enter peripheral circulation, where they live for a few days and, later, die by apoptosis. Thus, receptor editing, clonal deletion and Anergy in the bone marrow ensure that B cells leaving the bone marrow are tolerant to all self-antigens. In the next video lecture, we will study peripheral B cell tolerance. Thank you for watching.